Yeah, all right. So, yeah, thanks very much. It's great to be here and be amongst this, I guess, kind of new community. I'm meeting a lot of new people here. So I'm going to tell you kind of a different story and a shorter story, one that actually has no equations in it. So we're going to go on this chemistry adventure together. So I want to point out some of the big problems that exist in photochemistry and modeling these reactions in light promoted and excited state-based reactions. And we're motivated by these energy-dense materials. So this is, the Office of Naval Research code for explosive. And one of those is cubane. So cubane is kind of a beautiful compound to organic chemists. It's really, actually remarkably stable despite being explosive. It's pretty dense. It's got a lot of strain energy, which will mean something to the chemists. And it's stable at really high pressures. So this means you can really compress it and store it. Turns out you can stack cubes, right? Pretty well. Boxes stack pretty well. Okay. So what we do in trying to understand these reactions, right, to make things like cubane and other things that are strains, is we perform these what are called non-adiabatic molecular dynamics trajectories. So these are all quantum mechanical or quantum chemical based simulations, and we can watch molecules dance if you like once they're injected with 5 electron volts of energy. And if you give it a little bit of time, about 500 femtoseconds here, it goes off to form the cubane itself. Now femtoseconds, right, just calibrate ourselves on time scale, right, that a femtosecond is a 10 to the minus 15 seconds, right, so it's extremely fast. And these simulations are also really expensive computationally. So, to give you an idea, to run this simulation you see here, right, this is taking about a human month, right, or about a month of wall time, right, to get through, because calculating the energies and the forces on this molecule on a femtosecond by femtosecond basis, right, is really expensive because we have to use these multi-configurational methods. And I guess to give a more global view of photochemical reactions at all, the first thing that happens is you're starting down here at some minimum on a potential energy surface. I've put bond breaking and forming on the x and y axes just to represent kind of an axis, right, that a chemist would see as an important part of the reaction, and then potential energy on the z-axis. So molecule goes here from the reactant, gets excited with a photon, and it now has the same exact geometry in the ground state, but has an excited state electronic configuration. And as soon as you're in that configuration, then the molecule wants nothing to do more than to get down to the ground state. And that can happen through a radiative process, right? So it can find a kind of local minimum, like over here, and then fall down through a process called fluorescence, and you literally, light is given off, just like we can see colors. This is how that happens. Or it can go through this kind of peculiarity here called a conical intersection. And this conical intersection is a simple representation of a structure that has the same geometry and energy on both the ground and excited state surface. So this is very, very far away from equilibrium. And I'm pointing all this out because, organic chemists have a hard time, both computational and experimental ones, have a hard time predicting what will happen in a photochemical reaction because we don't really have a good sense of what these structures look like, right? What the actual geometries changing in time look like, because they're so far away from this equilibrium geometry. And there's usually very harsh gradients, right, away from these conical intersections, which can lead to a variety of different products. Okay, so because these calculations are expensive and because there's a general need for a better intuition about these reactions, my group has been involved in this space and has really inspired us to start working in this area. And cubanes are just one class of compounds. Now, why this is a good dynamics problem, especially for machine learning, is because just for the somewhat simple reaction, or seemingly simple reaction of this starting material going to cubane, you can get a whole bunch of different products and a whole bunch of different pathways, right? So just to name some, you could have this bond break and then kind of revert either to the reactant again or to this anti-version of the product. You can have all kinds of pericyclic reactions that we don't need to relitigate that organic chemistry horror that some of you might have gone through, and then you can get a whole bunch of different products down here too, where clearly these products are the preferred ones. Okay, so mapping out these different pathways can be done quantum mechanically if the molecule is small enough, if the active space is not too large, right, which we have done for this particular reagent. But we find that there are other examples of this reaction, such as one where this reactant, this tricyclooctadiene, is permethylated. And if you have the permethylation, if you basically put this outside in LA, right, 25 Celsius in light, you get a 1% yield. Wow. really impressive. But there are other things you can do. So you can use these trifluoromethyl groups. If you heat that up and add light, then you can get to this product in a much larger yield, right? 20%, but overall still, somewhat small. And then you can use this cyclopropyl group, which can also then make this fully photochemical and get to 50% yield to a really energetically dense material. So we were curious about understanding why this reaction, essentially doesn't happen for the unsubstituted parents, but there were varying yields for the substituted cyclooctadienes. So these calculations, as I've mentioned, are expensive, so just want to give some scale here. So for the parent, that is the one without the substituents, a single point energy, just one energy along the 2,000 energy points we need to run in a typical trajectory, takes about a minute or 5 days for the whole simulation. If we add methyl groups, this number gets really high, really fast, right? So we're at 2 years almost, right? It's like maybe a short master's, just to do one reaction that also doesn't have a guarantee of leading to products, right? If this can run in a picosecond, it'll take 2 years. Maybe you'll get some products, maybe not. This can get worse with trifluoromethyl groups, right? So now 2.6 years, and for that largest substituted TOD, we're talking about 5.5 years, right? So this is not going to happen. My students would all walk out, and I would be, without a single grad student in my group. So we had to come up with a machine learning model that would work and allow us to make these predictions faster and more efficiently towards making this important chemical discoveries about these reactions. Okay, so this is a postdoc in my lab, Jingbai Li, who helped me to create the pyramid code. So the pyramid code essentially is a machine learning photodynamics simulation code that helps us push the limit, right, from the 1 picosecond, 10 to the minus 12 seconds, all the way into the tens of nanoseconds, if not hundreds of nanoseconds, which is totally transformative, right? I mean, there's many organic photochemical reactions aren't complete in 1 picosecond. Many of them have low quantum yields. So being able to simulate out to tens or hundreds of nanoseconds allows us to simulate virtually any photochemical reaction that's happening on the singlet manifold. Another thing that this code helped to do is to move this field away from just model systems, right, that underwent remarkably simple reactions, and instead look at really complex reactions that also invoke some stereo selectivity as well. And the way it works is pretty straightforward. So we represent the molecules through a Coulomb matrix or an inverse distance matrix. So it's just every distance is 1 over that. We feed this to a really standard MLP, and from that we can predict the energies and the forces, which is basically what we need to propagate these trajectories in time. And we're able to get in energies well within the chemical accuracy of 1 kcal per mole. These neural nets are differentiable, so we can get forces from there. And we generate the training data. So this is really important here because the search space is it's not infinite, but it's pretty close, right? There's many different possible configurations that these atoms can be in different geometries. So one of the ways that we do is just a normal kind of Wigner sampling, if you're familiar, that samples the geometries within a local reactant or product, okay? We also do a geometry interpolation, so we find those crossing regions, that part that I showed you earlier, and then that allows us to find more of the points that are in between, right, the surface. And finally, we can use the trajectories that essentially were being run that may or may not have been useful for understanding the reaction. So taken together, this gives you a really good way to introduce some solid chemical human bias into this, right? Because if you want to see a cis-trans isomerization, that's exactly what you'll see, because that's the conical intersection you gave. So we do also an active learning approach called adaptive sampling that helps us to find parts of the potential energy surface that are undersampled, right? And then just essentially we take those new points that are not sampled, we recalculate them with CAS, retrain the neural network, and then repropagate the neural networks. And that's helped us to map out some really complex dynamical processes. So this is just to give you one example, as I'm sure I'm maybe close on time. This is one example, right, starting from this permethylated tricyclooctadiene, and all the different products B, C, D, E, and F that could all be formed, right? And this is only possible to map out all these pathways because of the adaptive sampling that helps us to find these define these different pathways in real time as the training is happening. And we've been able to reproduce the experiments actually remarkably well with quantum yields. So the experimental yield is 1% for the cubane. For the per methyl, we get exactly 1%. A wild coincidence, so don't think we're that good. We're pretty good. And we map out all of the different pathways that are shown experimentally. What's kind of interesting too is if you increase the steric bulk and put the trifluoromethyl group, in other words, add fluorines instead of hydrogens, we see that a lot of the trajectories that were in this region over here, just to show you before, right? All these trajectories here are now gone, and that's because there's substantial closed shell repulsions. These are kind of steric clashes, if you like, between these fluorines at the conical intersection that leads down that pathway. So that helps us to understand that some of these trajectories essentially are being blocked because of these steric repulsions and funneled more towards the cubane products that we actually want to see and experimentally are observed. And I want to just kind of close with one more slide and say, it's not enough to just block a pathway, right? Like you might on a thermal reaction or a ground state reaction. You also have to tilt the potential energy surface to favor the product that you're looking to do. So what we see with these substituted groups, these are non-covalent interaction plots. Don't have enough time to talk about them, but green means good and stabilizing, red means bad and destabilizing. So you can see by just adding these methyl groups, there's considerably more green dots. This gets even more exaggerated with the trifluoromethyl groups, as well as some other kind of fancy stereoelectronic effects that I probably also don't have time for. And same thing with the cyclopropyl groups. So overall, these substituent groups are helping to stabilize the pathways and while also blocking the disfavored pathways. So thanks to funding agencies and for the invitation, and I'd be glad to take any questions.
Yeah, this is super cool.
Thanks.
Couple quick questions.
Sure.
The first one actually on your last slide, I think, or
this one?
Yes. Why is only the bottom one red, or like the bottom lobe red versus the other ones? They look.
You mean this?
Yeah, and then is it not symmetric or?
I didn't think about that, but
okay, yeah, maybe I'm just.
This is probably just sigma bonds.
Something, yeah.
Clashing, yeah.
And then my other question was, so I'm very interested in how ligand, basically choice of organic ligand, I'm interested in hybrid organic and inorganic systems can help, encourage the formation of strained inorganic structures. So this was like very exciting for me to see. I guess from your experience, what's your kind of intuition for the types of ligands that make strained structures more or less favorable, and would that intuition generalize to beyond covalent or like not? I mean,
right.
Like
all
you get
organic.
the question.
Yeah.
Right. Thanks.
I mean, so I'm cautious to give any big generalizations, mostly because so much of photochemistry, right, is just not generalizable right now. We have like a series, right? There's 4 reactions, right, and each one, and we have to train the neural net for each one of these, right? So, I think if we have a series of related ligands, we might be able to draw some of those conclusions. Especially if there's anything kind of light activated about it. Yeah.
Cool, thanks, super cool stuff.
Thank you, yeah.
Quick clarification, did I get it right? So you need about 1,000-ish points to parameterize the neural network per...
So it's usually around, I think when we first started it was close to like 6,000 or 7,000 points. But then we started doing the active learning, which brought it down maybe to about 2,000 or 3,000 for the initial set, but we end up with about 5,000 or 6,000 after all the learning is done.
Any hope we can do a transferable?
Hope, yes. So we're on actually.
Maybe with fancy descriptors.
No doubt. We're on an NSF Institute grant right now, which it's the data-driven dynamical design (ID4), with plenty of machine learning people that are going to try and help us with that space, definitely. So, hopefully, yeah.
Actually, mostly just answered my question, but with when you were using just the 6,000 initially without using the active learning, how were you actually picking those points?
Yeah, so it was very, I would say, chemically inspired, right? So we essentially tried to map out the important mechanistic features of the reaction, right? So we'd sample around the reactant, the product, the conical intersection that would correspond to that inner conversion, and then the trajectory information. So it'd just be a mixture of these three, this kind of composite approach, if you like.
And when you went to the active learning approach, did it tell you that you were wrong? In other words, did it give you,
Yeah.
did it say you should have looked over here instead of there?
Yeah, what it I think told us was mechanistic pathways that we hadn't thought of, either because we're bad chemists or it's complicated chemistry or both. So one of those things was, so actually in this structure here, one of the pathways that this found, even for this kind of simple cis-trans isomerization reaction, was actually a 1,2 migration of one of the hydrogen bonds, one of the carbon-hydrogen bonds to lead to a carbene. An experiment, I thought that was maybe an anomaly of the active learning approach, but actually that is a pathway that's known, especially in electron-deficient alkenes like this. So we're actually learning, as chemists, a lot from these trajectories.
That's very cool.
Thanks.